Yep. So essentially the topic of what I'm going to talk about is really what can the immune system do against these reservoir cells? And just here I have some of my disclosures, right? So to be transparent. So I'm going to start by saying a few words about what these reservoir cells are. Just that's a recap. Most of you know this already, and then I'm going to go into some new technologies. So generally, I mean, we are talking about these reservoir cells all the time, and we know that this is a very small number of cells. That's really a big problem to understand these cells because there are so few of them. In many studies, it's 1 in 100,000, it's 1 in a million, sometimes 1 in 10,000, some in that range. So it's different in every person, but generally it's a very small number of cells, and that makes it very difficult to analyze these cells. And that's why we believe new technologies are, in many cases, needed to better sort of characterize these cells. We also know that these cells are very established at a very early stage in the disease process, in most cases, within a few days after viral transmission. And the problem is really that these cells are not being targeted or eliminated by the type of treatment that is currently available. And that's why these cells are essentially persisting forever, indefinitely, and they are sort of responsible for rebound that occurs when treatment is interrupted. Good. One question that comes up for these cells all the time is, why is the immune system not able to get rid of these cells, right? In all alternative viral infections that we typically know about, in some form, the immune system is able to eliminate virally infected cells, but HIV is the outlier in a way. Here, the immune system is failing in eliminating these cells. And that's why HIV is really one of the very few infectious diseases for which no curative treatment is currently available. So that's what I'm going to talk about later then, is why is the immune system not able to do it? Just the traditional explanation for this is that this is because of what people have been calling viral latency. So the cells that are infected are not really expressing any viral proteins, and the immune system can't identify these cells, and that's the reason why the immune system is ultimately failing. But the story seems to be a bit more complex, and that's what I'm going to talk about later on. So before getting into this, I just wanted to make a few comments about the technologies that we are using to analyze these cells. I believe there's a lot of progress in the last couple of years in developing better approaches to analyzing these cells. And what we are specifically using is the sort of next generation sequencing platform. That is an important tool for us. And if you look at the history of medicine, there's always important technical inventions that have transformed medicine. You could think about like sort of when the microscope first became available, that was a huge transformation in medicine. And I would sort of propose that next generation sequencing technology that has been developed just over the last 10 years maybe is maybe comparable to what the microscope was 100 years ago. We can now look at each single molecule in a cell in a very high resolution analysis, and that changes all the things that we can do. So the types of assays that we have developed here are sort of designed to look at different parts of the virus and provide different pieces of information to understand the biology of these cells as deeply as possible. The first assay is called what we use the terminology here is FLIP-seq assay. So this is an assay to look at the entire sequence of the virus. That is already different from what was done in the past, when sort of bits and pieces, small pieces of the virus were looked at and you didn't really know what else was going on. But now we have this ability to look into each single viral sequence that we can see and sort of analyze the totality of that sequence. So that's one part of it. Then we have a subsequent assay that we call MIP-seq. So that is an assay that is designed not just to look at the virus itself, but also to look at the chromosomal integration site. That's an important part of the story here, that HIV manages to integrate itself into the human genome. It's becoming part of the host genome. And the location where it is integrating is an important part of it. And I'm going to talk about this later on. Then we have these technologies to look at the viral sequence, the integration side, and also look at what we call transcription. So how much virus is actually being produced and how many viral RNA sequences and viral proteins are being produced. So that gets into this direction of what we call latency. So is the virus really silent in the sleeping condition, or is it not? And using that technology, we can actually address that question in more detail and get more information on this. And the last assay is an assay to look at the phenotype of the infected cell. So that relates to the ability of immune cells to target infected cells and to recognize them and to kill the infected cells. And because the number of infected cells is so small, there were no good technologies in the past to specifically look at this very small number of cells that are infected with HIV. And this new technology now allows us to look at that surface phenotype in greater detail, and we can sort of understand how do these infected cells look like to the immune system. So what all these assays have in common is that these are sort of high-resolution assay. They focus on each individual infected cell, sort of very high resolution. Then at the same time, we use direct material that is collected from study participants. So we are not doing any sort of in vitro infection experiments or animal experimentation or any of these things, but really we work very closely with study participants. We are collecting samples and we are directly analyzing these samples and the types of assays that we are using. And at the same time, these assays are designed to provide different pieces of information so that we can get sort of a holistic picture and impression of how these cells behave. And specifically, we have been doing, I'm going to talk about this later, but we have been doing longitudinal studies where we can understand what's the evolution of these cells and how do they change over time. Good. So I'm going to just go over each assay a little bit and talk about what these assays can do and what kind of new information we can get out of it when we use them. So the first assay is that full genome sequencing assay that by itself is designed to distinguish defective viruses from the intact proviruses. That by itself is a very important distinction. We know many of the viral sequences that we find integrated into the host genome are defective for various reasons. It's about 90% of the viruses, and only about 10% are genome intact and can potentially be replication competent and can drive rebound viremia. And sort of focusing on that small subset of sequences that is genome intact is an important distinction. And by sequencing the full virus, we can do this. Just going to give an example here. So this is one particular study participant that was analyzed here. So the way we do it is we collect a sample, a blood sample, then we extract the DNA, and then we do this PCR assays. And what we are getting at the end of it is sort of like a diagram that looks like this here, and the bottom part of it. I believe on the next slide there's a better example. So this is one particular study person here that was analyzed in great detail, and each of these horizontal lines that you can see here is one viral sequence that was identified in this person. Then we have sort of biocomputational approaches to distinguish now the sequences that are intact from the sequences that are defective. And you can see here the sequences that are intact are indicated in this color, and it's just a relatively small proportion of all the sequences that we can get. So these types of diagrams that's what our assays are producing, and we are sort of calling this a virogram, and it gives us sort of a good idea how does the proviral landscape, sort of the combination of viruses that are present in a specific person, how does that look like? So these are the ones that are intact, right? A very small number. And then we really feel this full genome sequencing is quite a bit of an advantage because it gives us a lot more information. An alternative assay that has been used in the past is shown here. So one is the total proviral DNA assay. So that was just checking on a small segment here. You can see this in this upper part of the slide. Mm-hmm. That was what used to be analyzed, right? And now we are looking at this entire viral sequence. So it's a lot more informative to look at the full sequence. There's also that IPDA assay that has been developed, which looks at two small pieces in the viral genome. One is here at that epsilon region, and one is in that envelope region. So looking at those two segments at the same time, but that is certainly an advance compared to the previous technology, but is still missing a lot of information about the viral sequences. So looking at each virus in its totality is really what we believe the most informative approach.
So looking just at this graph, I just want to make sure I'm understanding it. The dark blue lines, that's an intact virus.
Mm-hmm.
Just all of those dark blue lines, those are the ones that are capable of replicating. Everything else in here is not a replicating virus. All those other lines are defective viruses.
Precisely. All the ones, these horizontal lines are being sequenced. They are HIV sequences, but they have defects that preclude active viral replication.
Okay.
Yep. Yep. So we have optimized that technology quite a bit to a point that it can be done sort of large scale. We can generate from a study participant sort of like 600 sequences or so, and that's sort of the current standard. And we are always trying to optimize the technology to get more and more sequences from a study participant to get a deeper view. Yeah. So now I'm going to get back to that original question. What can the immune system do against the reservoir cells, right? And then the traditional answer to that question has essentially been always sort of nothing, just because of this part of what we call latency, right? The virus is hiding, it is sleeping, it is not transcribing. The immune system doesn't know which cells are infected and which cells are not infected. And for that reason, the immune system essentially doesn't have an ability to do anything against these cells. But we believe that perception is changing a little bit now. And we see signs of immune activity against the reservoir cells when we do two different types of assay. One is looking at the integration sites, and one is looking at the surface phenotype. And that's where the assays that I just mentioned are particularly useful for. So I'm going to talk about this next assay here, is that MIP-seq assay, which is that assay for looking at the integration site. And before doing this, I'm just going to talk a little bit about the background here. So as I said before, HIV really has this ability to integrate itself into the human genome, and that human genome is quite complex. And what we know so far about it is that it is large. It has 3 billion base pairs, right? So it's a really large genome, and it has 22 chromosomes, which are present in two forms. One is from your mom, and one is from your dad, and then there's 2 sex chromosomes, right? And it's been first sequenced about 20 years ago or 25 years ago by 2 different groups. And what we are benefiting from now is really that we have that genome available that is very well characterized and very well annotated, and we can really use that information for figuring out where HIV is integrating. One important observation that was made when they first sequenced that entire human genome was that only a small proportion of the genome is actually genes, right? So the genes are the one that sort of the sequences that are encoding proteins. And the initial assumption was that essentially that whole genome is full of genes, but that was not true. It's about only 20% of the genome that we have is encoding for proteins, and 80% of the DNA is so-called non-coding DNA. So that's when no protein expression is happening. So, and what's also interesting is that then HIV figured out strategies to specifically integrate itself into the genes. That was an important observation that was made maybe 10 to 15 years ago, that HIV has figured out molecular strategies that guides the virus into those genes. Why is that useful? Probably because being integrated into the gene is a benefit for the virus, because in these positions in the genes, the viral gene expression is most effective. So in these protein coding parts of the genome, when HIV is sitting in there, the viral proteins can be most effectively expressed. And sort of that is an important part of the background knowledge HIV wants to be integrating into the genes, because for the virus it is most beneficial to do it. At the same time, we also know that this is not a perfect mechanism. So there's always viruses that are not getting into the genes. They are going somewhere else. And that becomes relevant in my subsequent presentation here. So this is just sort of confirming what I just said. Here's a couple of studies that people have done where sort of cells were infected in vitro or in vivo, and different types of studies. And they looked at the proportion of integration sites that were in genes, okay? And you can see in the vast majority of studies that somewhat in the range between 80 and 90% of all the genes, of all the viruses are in genes. And only a small proportion of HIV goes into these non-genic regions, so the non-coding parts of the genome. So then we can use this assay, that MIP-seq technology, to be specific and figure out where are the genome intact proviruses, the viruses that have are replication competent, where are those located, right? And for this purpose, we have developed this complex technology here. So we are diluting DNA that we are collecting from blood samples from study participants, and then we can use that whole genome amplification process here. So essentially each single virus gets amplified by about 1,000 to 10,000 times. Then we have enough material to look at the viral sequence and at the same time also look at the integration site of that particular virus. So that helps us to distinguish where are the defective viruses located and where are the intact viruses located. So this is an example where we've done this in a study participant here. You see what we call a phylogenetic tree here on this part of the slide. Each of these sequences here is one intact viral DNA sequence that was isolated. And you can see here the location was precisely mapped, so we can see in what chromosome it is. We can also tell at what specific base pair location in that chromosome this particular virus is integrated in. And in the vast majority of cases here, these intact proviruses were integrated in genes, right? So when there's these names here, these are gene names. So for example, AIDA, House, TMEM116, and so on and so forth. So these are all the genes that HIV has integrated in. And that's how this typically looks like in study participants. So HIV finds its way to the genes and persists in genes. But at the same time, we also see that in some patients, it's quite different. So for example, here we have someone who is an elite controller. So this is a person who maintained drug-free control for about 20 years. And here it looks like this. So we have each of these dots here represents one intact proviral sequence. And you can see here this upper cluster of sequences, they are all 100% identical. So this is what we call a clonal cluster of sequences. And in this particular person here, it looks like there's only two different clusters of intact proviruses. And when we look at the integration sites here, then we find that these viruses are actually located at positions in the genome that are profoundly different from what we see in alternative people who are not elite controllers. So here, for example, this large clone is integrated right in the center of the chromosome, okay? You see that chromosome that has that typical shape here, and there's that central part, we call it the centromere. That's where the virus is integrated in this particular person. And what we know is that that central part of the chromosome is what we call a gene-free region, or like a gene desert. So there's no genes there, and there's no transcription and production of viruses possible at these locations. At the same time, we have a different cluster down here. So this is in a gene, but in a very special gene. It's a ZNF gene, okay? And these ZNF genes are located on chromosome 19. And what we know is that these ZNF genes are also very, what we call transcriptionally repressed. So they have specific chemical modifications at these locations that are actively inhibiting production of viruses from these types of sequences. So in this elite controller here, it looks like HIV is really in very distinct locations in the human genome. It is at positions where viral production is not really possible. And the terminology that we use for this is also the sort of the virus is located in block and lock positions. So it can't really be expressed. It's a dead end for the virus. And we are proposing that if you have this particular location of the intact viruses, then that is contributing to the ability of maintaining drug-free control, because the HIV is sort of locked in positions in the genome where it can't express itself anymore. So that's sort of the idea behind it, but we wanted to be more specific then and really look at what can HIV be expressed or not at all. So is HIV really in this latent position or not? And for this purpose, we developed that next assay here, which is called the PRIP-seq assay. So this is an assay that allows us to look at the HIV sequence at the integration site, and at the same time also at the production of viral RNA, which would lead to viral proteins. Okay, so that really addresses the question, can HIV be expressed, or is it in that sleeping condition in a sort of latent condition? It's very technical here. I'm not going to go into all of this, but we start with material that was collected from study participants. Then we are isolating the DNA, we are isolating the RNA, we're getting the proviral sequence, the integration site, and we are also measuring the number of viral transcripts, so the HIV RNA that is being produced. And all of it we do from a single infected cell, right? So that's a very complex assay here, and really allows us to address the question, is HIV really latent or not? Is it really transcriptionally silent? Is it sleeping, or is it not sleeping? Okay. Mm-hmm. So we've done this here in a study where we focused on 701 individual HIV infected cells, so a large number. Then we looked at how many of these cells are actually producing viral RNA. And this is sort of in samples is collected straight from patients, from study participants. So it's not in any way manipulated. It's really telling us, is there any viral transcription happening? And the surprising finding here was when we generated this data is that many of the viral sequences are actually not in the sleeping condition, are not latent, right? So here between 30 to 40% of the viral sequences that we analyzed were actually actively transcribing viral RNA and making viral proteins. So that was sort of a little bit violating that sort of dogma that we always thought about, that HIV is sleeping and the immune system can't recognize it, it's latent, and it doesn't seem to be true if you use these types of technologies. It looks like there is some transcription going on in some of these cells. It depends on the chromosomal location, right? So we were able to distinguish this here, the proviruses or the HIV sequences that are sitting in genes and actively transcribed genes are in many cases transcribed and not silent. And then we have these sequences that are in non-genic positions and non-coding parts of the genome. Those are the ones that are really repressed and are in sort of this block and lock condition where no viral transcription is possible. So I just wanted to get back to this idea of latency because that's so important. And I believe a lot of people are talking about latency at your meetings, right? So, I mean, we are always thinking about HIV as being latently infected, but what I just told you is that HIV in many cases integrates into the transcriptionally active, so the protein coding parts of the genome. So that doesn't really make sense that HIV should be sleeping when it's integrating itself into the parts of the genome that are the most active ones, right? So that's one part of it. Then we know that the drugs that we are using for treating HIV, they are not actively blocking HIV from transcription. So it's not that the drugs are putting HIV to sleep. Yep, so that's one part of it. And then we also know that when HIV treatment is stopped, then the virus comes back in a couple of weeks. So that also argues against this idea that HIV is sleeping all the time, because we know it comes back pretty quickly. So to some extent, we believe that this latency idea is not fully true, and there's actually a pool of reservoir cells that is not latent, but it's actively transcribing viral genes and is making viral proteins. And we believe that's actually, in a way, it can be interpreted as good news, because that gives the immune system a chance to recognize at least some of these cells and target some of these cells. And we are seeing evidence for that in the work that I'm going to show you now. So here's
can I just interrupt? I want to ask.
Absolutely.
So latency, would we say that that's simply not a true idea, or is it that some things are latent, but it's not nearly as large as we think? Like, is latency totally not true, or is it a little bit true, just not as big as we thought it was?
Yeah, it's a great question. So I would say an important part of this is that extreme diversity, right? So we have this huge pool of reservoir cells in each study person, and some of them might be in this true latent condition, some of these cells, but some not, right? And I believe how many of them are truly latent and how many are not, that is changing over time, I believe. That has to be viewed in the context of what the immune system can really do against these cells. Some of these cells that are maintaining some viral transcription and are not latent, those are the ones that the immune system can recognize and can probably target. And that's why the fraction of these cells is likely to be decreasing. That's at least what the studies show. So, but it's a great question. So here we try to address it by looking into this particular study participant here. That person was identified back in the 1990s, started ART at that time, and then sort of remained on suppressive ART for pretty much 20 years, and had no viral blips, but good immune reconstitution. And it gave us an opportunity to sort of see what can the immune system do against these reservoir cells over these very extended periods of time. It might not be very effective what the immune system can do, but some of the cells that have ongoing viral transcription might be particularly vulnerable. And sort of to look into this, then we did that integration site analysis, and I'm just showing a few examples of people that we have studied for these very long periods of time. What we're generating then are these types of diagrams here. Each symbol here reflects one genome intact viral sequence. And you can see here in that one study participant, LT03, we only find one really large clone of intact viruses that have all the same viral sequence and are all integrated into the same location in the genome, which in this case is chromosome 18, and it's right in that centromere location. So that's that location that I talked about earlier. It's really that region of the chromosome where no protein coding genes are located, where no active viral transcription is happening. It's that block and lock position where HIV is sitting. And we don't find any alternative intact proviruses that are sitting anywhere else.
So what causes that? I mean, that sounds like the $64,000 question. How do we get that to happen in all people? Or, is it that, I had a question initially about all of the different places where it goes. And is it a characteristics of the virus itself? That, like you say, that some are latent and some are not? Or what makes it go to certain areas and others? I mean, why is it attracted to some genes and not others?
Yeah. No, it's a great question. I mean, we believe that centromeric location here is something that HIV doesn't like at all, right? Because it's a block and lock position. So HIV tries to stay out of that region as much as it can, but just by random effects during that initial period when the reservoir is seeded, some of the viruses go into that centromere region. It's a very small fraction. We typically don't find it at all because there are so few of these sequences going into the centromeres. And these are the outliers, right? So usually we don't see them at all. Just after 20 years of treatment, we believe these initial outliers sort of have a benefit because they are the ones that are surviving up to this point, because those are the sequences that the immune system really cannot see because they are not expressed, they are blocked and locked. The alternative sequences, right, that sit in regular genes where some degree of viral protein expression is possible, those are the ones that the immune system has a chance to recognize and to target and to eliminate. And that's why we don't see them anymore after long periods of time. It doesn't mean that all of them have been eliminated, right? That's the limitation of our current technology, that we can't look at all intact viruses. But we can certainly say that the immune system has been active in eliminating a good chunk of the viruses that are sitting in genes where some degree of transcription and viral production is possible. And I'm suggesting that because we looked into a couple of these people here who have been on treatment for a long period of time. So for example, LT01, you see a very similar pattern here, right? One large clone, again, integrated into that sort of pericentromeric region, in this case on chromosome 21, and an additional sequence down here that we found that was on chromosome 17. So it's, in a way, it's remarkable that HIV can only persist at these very specific locations. We always think of HIV as being very flexible and being able to do all kinds of things, but when it comes to persistence, long-term persistence in the setting of ART, it seems like HIV doesn't have a lot of choices and can only persist at very selected parts of the genome. Here's two more study persons that we looked at in detail, and again, we find that HIV is persisting at these very atypical locations in non-coding parts of the genome, like non-genic regions, or in these ZNF genes here that I mentioned before, which are also genes that are repressing the virus from being transcribed. Again, I mean, the technicians and the students who are doing this type of work, they always come into my office and say, this is so predictable in a way, right? HIV, after 20 years of persisting in the human body, it really is only limited to these very atypical non-coding parts of the genome where it can persist.
So it's really not a host factor. It's a factor of HIV being attacked years and years under antiviral pressure, right?
Yeah, that's exactly right. So HIV is persisting. It's getting exposed to the immune system for many, many years. And it might not be super effective what the immune system is able to do, but over 20 years, it sort of builds up, it accumulates. And what we are seeing then is that the immune system is able to select for certain types of viruses. And that gets back to that initial question that I was bringing up, right? We always thought the immune system is powerless against these cells, but in reality, it's actually quite functional and getting quite a bit of work done here. So because
it's been seeing 30% of them for a really long time.
Yeah.
Wow.
That 30% might not be sort of, we don't know exactly. It could be more, it could be less, and so on. Wow. I'm just going to try to relate this to one particular story that I wanted to share with you. So this is a study participant here who was participated in a study in Denmark. He was getting, as part of that study, getting romidepsin, which is a latency reversing agent and a broadly neutralizing antibody, and then was started on ART as well. And in this case, he remained on ART for about 400 days and then interrupted ART and then maintained this really very impressive form of drug-free control going on now for what, like 5 years or so almost, right? And you can see one blip maybe here, but all these other viral load tests have been negative all these years. So this was an opportunity for us to look a little bit more in detail what is going on here in a situation like this. And essentially, we sort of did that MIP-seq assay that I was just talking about, and we generated this type of data here. So it's a little bit complicated, but at the day of ART initiation, we have a large number of intact viruses that we analyzed, and almost all of them are in genes, right? So in coding parts of the genome here, right? So that's, as I said before, that's where HIV likes to sit. That's where HIV is integrating itself into. But over time, then it's changing, right? During the time of that treatment interruption period, we see that all these viruses that were sitting in genes are essentially gone. We can't see them anymore. And our hypothesis is that they have been eliminated by the immune system. And then what's left over are only sequences that are sitting in these sort of centromeric locations of the chromosomes. And you can see here, we found at least 4 different types of clones that are sitting in these centromeres. One is here in a chromosome Y position. Then there's one in chromosome 4. There's one in chromosome 9. There's one in chromosome 19. and one more in chromosome 4. So again, it shows that situation that HIV, when it comes to persistence, it really doesn't have a lot of choices, and all the viruses that are sitting in genes have apparently been eliminated here, and what's left over are only these viruses in the block and lock positions. And we are proposing that that's really what it takes to get to this post-treatment control status. If you have a reservoir where HIV is only in block and lock positions, then it's sort of game over for HIV, and there's no ability to have rebound anymore. That's the hypothesis. I want to be critical, and I have to say we are working on a larger number of study participants to confirm this and make a stronger case here. But it's sort of reflecting, in my opinion, sort of that ideal scenario. In the best case scenario, it happens like this, right? The immune system is able to kick out a ton of viruses that are sitting in the genes. Those are gone. And then only the block and lock viruses are persisting. So for the last part of the talk, I just wanted to briefly focus on that phenotype aspect of it, right? I mean, one question is, why is that selection process that we are seeing here, why is it not so effective? Why is the immune system not able to do a better job here in everybody? It seems to be going on in certain highly specific people, but it's not happening in the majority of people. So what's the reason for that? One way to look at this is, as I said before, is to look at the phenotype of these cells and sort of understand what is the immune system seeing of these cells. And we have developed here this complex technology, again, that is all very technical. I'm not going to go into all of it, but we have the ability now to use more than 70 to like 100 different antibodies that we can use to stain these cells, to label them, and to see what type of surface markers they are expressing. And then at the same time, we are putting them into a special instrument that allows us to figure out if this is an HIV-infected cell or a non-infected cell. So we are going through thousands and thousands of cells, and in each cell, that technology allows us to determine if viral DNA is present in that cell or not, okay? And then we can sort of get that information on the surface phenotype, and we can see the cells that have HIV DNA in it. How do they compare from that surface phenotype expression perspective to the cells that are HIV negative? So it's pretty complex. It's but really in a way showing the power of the technologies that are available these days. And this is just an example of how this looks like here, right? So we are looking into large numbers of cells. Each of these dots here is one cell, right? So in this particular study person here, we looked in about 150,000 cells or so. And the HIV-infected cells are the ones that have these bigger type of dots here, right? And here in this type of the slide, we are looking into all HIV-infected cells, but here we are only looking into the HIV-infected cells that have intact, so the replication-competent viruses in it, right? It's a very small subset. Again, it's 150,000 cells that are being analyzed here, and maybe 100 to 150 or so are the ones that are HIV-infected, so... small minority. But what comes out of it, if we are using that assay, is then that in many cases, these types of HIV-infected cells have surface markers that are actually blocking them from getting killed by the immune system. That's particularly true for these markers here, HLA-E and PD-L1 and PVR. These are surface markers that are telling the immune system, don't attack me, don't kill me, and sort of are protecting these markers, right? And we believe that's part of the problem, that the cells that are surviving, these reservoir cells that are surviving for a long time and to some extent resisting immune activity, those are the ones that express surface markers that are sort of protecting them from immune recognition and immune targeting. And that is, in a way, it's telling us that these cells are under immune selection pressure in a way, because only the cells that do express these markers can survive long term. So you could interpret this as a sort of as a sign of the power of the immune system. But at the same time, it also contributes to the ability of some of these cells to persist for a long time and resist immune elimination. So I'm just going to wrap up here with the technologies. So, I mean, we always thought about the reservoir as being latent, but in reality, not all of them are latent, and some seem to be actually very vulnerable to immune recognition and immune targeting. And that's sort of a good sign. The host immune responses are not particularly effective in a lot of people. And that really gets us back to this original question, what can we do to make this process more effective, right? We see clear evidence of immune activity against the reservoir cells when we look into people who have been on treatment for 20 years, but we want to do it faster and we want to do it better. And that's a lot what my lab here and a number of people are focusing on. How can we improve that immune targeting of the reservoir cells and accelerate that selection process and better understand the mechanisms that allow the reservoir cells to resist immune-mediated killing. Here's just a sort of a summary of the types of like the collaborators and the team. And I just wanted to use that opportunity one more time to say thanks to your group in particular and for this great support from the community for the work that we do that is so mission critical, and we really appreciate that.
Wow, that's all I can say. I keep saying, wow. I feel like an old hippie. This is amazing. Well, I guess instead of me continuing to ask questions, we ought to go to the general format, or I'll take up the whole time. I'm so excited. So what do we got? Michael, Richard in the chat? Not much, right?
No, we had lots.
Okay, I don't see
Do you want me to do it, Michael?
Yes, would you? That would be great.
Sure thing.
Yeah,
So I
that would
think
be
first
great.
up, I don't know, Mark Gilbert, if you want to come on and ask your question about longitudinal follow-up, or
well, sure. In the first section, you were talking about using the historical data for a longitudinal study. So I'm under the impression that your ability now to get down on a molecular level is different than what it might have been 10 years ago or 20 years ago. So how do you do a longitudinal study when the data from then would be incomplete? Or was the data always there and we just didn't know what it was?
Yeah, no, it's a great question. I mean, the technology has really changed so much in terms of what's available now and what was available maybe 10 years ago or so, right? So that's why I believe it's really important to use the most advanced technologies that are available these days for analyzing samples that were collected many years ago. So that's really, I believe, a benefit of the collaboratories, specifically the work that Steve and others have focused on, like sort of collecting samples longitudinally over decades, really, and then using them right now for the types of studies that are being conducted right now. I can just say one example is that we worked on recently is a study participant who started ART 28 years ago. That's the longest one that we have analyzed so far, and sort of we had samples available from 28 years ago when ART was first started, and I'm sort of looking at it longitudinally now.
Thank you. Hopefully the next one is a quick one and kind of easy. Melanie Reese was asking if you'd be willing to share a PDF of your slides afterwards.
The slides, yeah, sure. I'm happy to do that. Yeah.
Excellent. Thanks.
Awesome.
Anna, would you like to come on and ask a question about activation and inflammation? So it was a question just, I think, maybe about HIV transcription and whether that contributes to immune activation and inflammation.
Yeah, because since the immune system remains active, so my question was that all we talk about all this immune activation and inflammation that contribute to all the comorbidities and so on that we see in long-term HIV infection. Has something to do with all you presented?
Right. So that's part of it, right? And it's sort of in a way contributing to this idea that latency might not be fully true. We know that people who live with HIV have abnormal levels of immune activation. That's been shown over and over again. And one reason for this is probably that HIV is not latent, right? It is not in a sleeping condition all the time. It is being actively expressed, and viral proteins are being produced, at least by some of these viruses, and that is contributing to immune activation. And it's also interesting if you look into some of these elite controllers where HIV is sitting in the block and lock positions, right? So you would argue HIV is not supposed to be expressed, but in some of these elite controllers, there are still higher levels of immune activation. And one of the reasons could be that in these elite controllers, you still have a lot of these defective viruses, right, that I talked about. So they are not replication competent, but viral proteins can still be produced to some extent from defective sequences. And these proteins that are produced from the defective sequences can contribute to immune activation. So that's why it's not just about the intact viruses. Those are the ones that are relevant for rebound, but for immune activation, it's probably a combination. Both the intact and the defective viruses can be important.
Okay, thank you.
Ezekiel, I see you have your hand up. Did you want to speak your question?
Sorry, I didn't mean to put my hand in.
Oh, okay.
Okay, thank you.
But
Thank you.
when I have the chance, I wanted to ask, did you also look at cells in the tissues, like in the gut or in, yes, or did you just look at cells in the blood?
Yeah, no, that's a fantastic question. So most of the work that I've just shown you today is really samples that were collected from the blood. That's just the most accessible samples that we can get. However, we have expanded efforts to look into tissue cells, specifically in samples from lymph node biopsies and also in samples from the gut, sort of from colonoscopies. We're actually expanding it right now to get more colonoscopy samples, which are so informative and so precious. We also had collaborated with the last gift cohort and have looked at samples from autopsy sample specimens. That is also quite interesting, and the experience so far in the context of the autopsy studies has been, I mean, we published that last year. We looked into 14 different types of tissues, so GI, GU, brain, lymph nodes, liver, and so on and so forth. And what was interesting that in most cases we actually found these large clones that I talked about, large clones of intact viruses, that they are sort of disseminated all over in the tissues. So we don't find much evidence that one specific virus is only present in one particular tissue. So it's more like we find them being present in all kinds of tissues.
Supercharger, did you want to speak your question? I know it's one that probably a lot of people have. If you're not able to, I can just read it. So Supercharger.
Yes.
Oh, go ahead.
Yes, I got disconnected when he was still talking about the Denmark patient. Did I hear that Lizepsin and Art and Binabs made him off treatment for 5 years? If it is true, don't you think we already have a remission which we should explore more, especially in Africa?
Yeah, so it looks like in this particular person, you could certainly call it a remission, right? I mean, that's a little bit comes down to what kind of terminology you are using, but someone who had sort of 5 years off ART and one single blip of, I believe it was 70 copies, and that's it. All these other measurements have been undetectable viral loads. So that is really, really remarkable. And it's not very common, unfortunately, right? So we only find this in very selected study participants, and we don't know exactly what the contribution of the romidepsin and of the broadly neutralizing antibodies were in this particular case. Was that really due to the study medication, or was it related to some alternative immune process that we don't fully understand at this point? It's hard to say it. In all fairness, I have to say in that study, there were additional people who also received the same study medication. They also received romidepsin and broadly neutralizing antibodies, and they didn't have that post-treatment controller condition. So it's certainly not just these two agents that caused all of it.
That's really important.
Patricia was asking in the chat, how is the virus directed to their various sites of integration? AKA, why in one case will it go to a centromere versus a very transcriptionally active region?
Oh, that's a very good question. Thanks for bringing it up. I mean, it gets very much into the biology of HIV. So in a short version, HIV has sort of adjusted very much to humans, right? That's something that we always have to sort of consider in these types of discussions. HIV has been living with humans and with monkeys for many years and figured out molecular ways to support its own life cycle. So in this particular case, HIV typically binds to a host protein that sort of acts as a travel guide and gets it right to the genes, right? So there is a specific molecule that HIV, the sequences can bind to, and that host protein is designed to bind physically to the genes and makes sure that HIV gets into the genes. That's what's happening in the vast majority of cases, but there's also always sort of outliers and failures of that mechanism in a way. And then we see sometimes HIV getting into these centromere locations, but again, this is an absolute outlier, right? So it's estimations is like it's sort of in one in maybe a couple thousand proviruses end up in the centromeres. That's sort of the failure rate, and the vast majority of them end up in the genes. It's very interesting biological principles, and it's also, I mean, doing this research with HIV is always teaches us how smart this virus is. It figured out very good mechanisms to get where it wants to be in the genome.
Back in the day, that used to be the question: is it really stupid or is it really smart? And you've given us the answer, I think.
Yeah.
Ezekiel, I'm sorry. I know you had your hand raised. I still see it raised again. Would you like to ask your question?
Okay. Thank you. I think this was really informative updates, but maybe after these explanations on evolution, might we think of some implications of viral diversity of HIV with respect to these response?
Diversity in terms of you mean sequence
viral
diversity or
clades,
different types of clades? Oh yeah,
different clades. Yes,
yeah,
different
clades.
Great.
So that's an interesting point, actually. That Danish patient that I presented is actually not a clade B person. I believe is clade AE, right? So a very different type of sequence, and
maybe that's why.
Potentially, right? I mean, there is that bias that the vast majority of studies have been conducted with clade B sequences,
B or
and
C.
we just don't, yeah, or C,
Yes.
right? And we just don't fully appreciate how clade diversity contributes to this. We're trying to expand this now, and are really making a sincere effort to include various different types of people and people who have different are living with different types of clades of HIV, to include all of them and study them in great detail to get a better sense how these yeah.
And I really asked the question because I mean, I'm from Cameroon, and in Cameroon we have a very wide genetic diversity. And from what I've observed, we don't have much of. We do have some elite controllers that I've observed some years back, but we don't have many patients controlling. And I was curious to know if these might be maybe due to that high genetic diversity.
That's why it's a really good question. I'm not sure exactly, but sometimes you could also argue the fact that you don't identify a lot of elite controllers could also simply be a diagnostic bias, just because these people don't become symptomatic, they don't seek medical attention, they are not being identified as much. So that could be one component of it. But it's certainly true. The different viral clades might be acting very differently, and to what extent the reservoir is better or is able to persist better or worse, that might very much depend on the different types of clades. So yeah.
I know also Siggy was asking about, did you look at viruses that were in tissues versus in the blood? Did you do things like that?
Yep. I mean, we did the autopsy studies, and we're doing lymph node biopsies and GI samples. So it's not easy to get those samples, right? And we absolutely want to make sure that there's no like there's no safety issues with collecting those samples. But at the same time, it is absolutely important. That's sort of, I would say, is one of the biggest limitations that we have so far, that the vast majority of work that we do is on blood samples, which is not the full picture and is hiding some information from us.
So, Michael, do you want to go into the next segment? Give us the last half an hour. Do we have any more questions?
I think we do.
Well, then do that first then.
Richard, did you see any other questions that we didn't get to?
So there was one from Aud. Sorry, if I'm not pronouncing your name correctly, Audi, about HLA selection. Do you want to come on and ask the question? It was just asking if you've looked at whether HLA class 1 or 2 might correlate with integration sites.
Also, good question. I mean, we don't really know what type of immune component is driving that selection pressure that I was talking about. So there's certainly a possibility that T cell mediated immune responses, so HLA class 1 restricted T cell responses, could be responsible for targeting the cells in which some degree of viral transcription is still ongoing and possible. At the same time, the data that we see in many different studies suggests that it is not just the T cell response, and that very likely there's alternative components of the immune system that is involved into it. Most likely, innate immune activity is playing an important role. Innate immune cells can recognize infected cells, virally infected cells, not by MHC class 1 or by HLA class 1 restriction, but by alternative mechanisms that are not fully understood at this point. So we don't find just to answer that question, we don't find a very strict association between HLA class 1 alleles and integration site profiles. It seems to be more complex, and different types of immune activity might be involved.
And Richard, you had your hand up.
Yeah, I just wanted to follow up on Patricia's question and ask, is there any possibility that where the virus integrates in the genome is telling you something about what the cell was doing when the virus was trying to integrate, like in terms of what genes were sort of active or might be available, whether the cell was trying to differentiate into something?
Yeah, it's a great question. I mean, what we know is that mechanism that guides HIV to the genes is very effective in a way. It guides HIV to the genes that are most actively expressed in a given cell. So that's really interesting in a way, that HIV has figured out just the cells that are most transcriptionally active and most effectively transcribed. Those are the ones that HIV is predominantly targeting and most easily able to integrate in. So it depends, right? If there is a cell in which a certain set of genes was active at the time of the infection, those would be the genes that would be most effectively targeted and most likely to harbor HIV.
Anything else, Michael, Richard?
I don't know. Did we ask this, Siggy? I don't know if we asked Siggy's question. We should be able to stop therapy in people who are 20 plus years on ART and see a lot of post-treatment controllers. Is that a fair assessment?
Yeah. So the limitation is that we only see a snapshot of the true reservoir, right? So what we see in our assays, we are not claiming that is the absolute truth. And that's the limitation of the current work. We have these high resolution assays and we see a lot of the reservoir that we weren't able to see before, but there's still stuff going on that we are not able to capture with the technologies that we have so far. And I would guess, what we are currently seeing is probably maybe the tip of the iceberg, the top 20% or so, and what's going on below the surface, below the sea level is just cannot be analyzed. I believe in certain selected situations, we are actually approaching certain study participants and proposing a treatment interruption, and we hope to be able to talk about this at some of the meetings in the future. But we certainly don't recommend it as a standard procedure, and we believe it only makes sense in very specific situations where a very deep analysis is possible, and we are very certain that to a large part, the virus is sort of in these block and lock positions.
I mean, yeah, we really need to be clear about that today because, I mean, for all the reasons Mathias said, but when we're talking about people who could get sick, people, people who could transmit HIV to other people. So, in other words, not ready for prime time.
Right.
But still standing everything else on its head, Mathias. I love it.
Supercharger, your hands up.
Yeah, this has been an amazing presentation. Thank you so much. My quick question is, have you tried to link up with the African researchers so that this kind of study be done in the African setting, where I really think we can get amazing results, much better than that you are getting from the Western world?
Yeah, no, that is really such an important question, and we have been trying to do this as much as we can. So we are very actively collaborating with investigators from Botswana. We are particularly interested in people who are living today as teenagers and young adults, but were acquired HIV at a very early stage in infancy, in many cases sort of at birth or immediately after birth, and sort of started treatment at a sort of in early infancy and then have remained on ART for maybe 20 to 25 years now. There's a whole generation of these types of people, just because prevention of mother to child transmission was not easily available at this time and in all parts. So this is sort of that young teenager and young adult generation of people that we are very much focused on, and trying to understand what can that pediatric immune system, what can that immune system do against the reservoir. There's reasons to believe that that pediatric immune system is actually better than the adult immune system. You see that in all kinds of diseases, COVID and things like that. So there's, from my perspective, a possibility that the pediatric immune system is doing a better job in selecting the reservoir cells, and that could also be at one point could be clinically significant at one point. So that's what we are very much studying.
So Mathias, what?
Go ahead. Oh, I think we lost Linda. Well, let me ask you, this is a question as well that came up. Someone wanted to discuss the brain compartment and also the ways to direct virus to the block stop mode. Thinking also about how endogenous retroviruses have incorporated into our genomes.
Okay, so lots of questions. So the brain is actually very difficult to study because it's not easy to have brain samples, but we have in autopsy samples we have been looking into the brain, and the most recent data show that indeed there is intact viruses that can be identified in the brain. It's relatively infrequent if you compare it to alternative tissues, like for example lymph nodes or gut tissues. We find relatively small numbers of intact viruses in the brain, but it's certainly detectable, and the proposed mechanism is that infected CD4 cells can sort of penetrate the brain from the blood and sort of infect the brain tissue by this mechanism. In terms of the endogenous retroviruses, that's also a really interesting story from my perspective. I mean, the endogenous retroviruses somewhat made it into our genome millions of years ago and have been able to persist in our genome. It's about 8% of our current genome is endogenous retroviruses, so quite a bit. And what's interesting is if you look at the locations of the endogenous retroviruses, right? So that's a little bit like similar to HIV. And we find that the endogenous retroviruses that have persisted up to this point, up to today, right, are also located in chromosomal locations that are sort of have block and lock site features. So you could argue there has been a selection process. The endogenous retroviruses that are sitting at very active parts of the genome have been eliminated over time in human evolution. And what we are living with today are sort of a combination of endogenous retroviruses that are located in positions where they don't cause a lot of problems and a lot of disease, and that's why we don't care about them as much.
We also had this proposition here from Siggy. So imagine a patient who is developing T-cell lymphoma at the time of infection. Would the HIV infection probably help in clearing the lymphoma because these cells are very actively replicating?
Well, it's not an easy question. I mean, I understand what you're saying. So you believe if HIV is infecting that clone would be more immunogenic. The immune system would be able to see that clone and would be able to target that clone. Is that about what the idea is? It's
I believe so.
I guess it's possible, but that has not been studied a lot. There's not that many people who have T-cell lymphoma, right? That's actually an interesting part of HIV. Is there's a similar retrovirus. It's called HTLV. It's somewhat related to HIV, and that is a virus that actually causes T-cell lymphoma in about 5% of people who are living with that disease. And HIV seems to be different in a way that it doesn't cause that particular lymphoma. So there's not a lot of research that has been done yet to specifically look at people who have T-cell lymphoma in HIV patients.
And someone else has asked can people who are living with HIV outside the US take part in your research?
Oh yeah, absolutely. So anybody can email us all the time, no matter where you live, and we can sort of figure out what the best program would be for someone to participate. And we have collaborators in Africa, in Latin America, in Europe, in Asia. So there's certainly a lot of opportunities to participate, and we really appreciate every single person who is willing to participate.
So all they need is your email.
Yeah.
Excellent. Excellent. So I just wanted to point out to people, first of all, there was the poll in there. If you wanted to take a chance to answer the poll question is still going up. I just saw another question. Likewise, are you aware of anything like the Last Gift study outside California?
So the biggest program is certainly at the University of San Diego. They're having this great program. At the same time, there's also a smaller studies going on here in Boston. So if people who are interested could also contact us here and participate in a program here. I'm not sure if there is a lot more centers who are having open protocols. I believe in Canada, there's some similar research projects going on, but I'm not 100% positive.
Jeff might know. Jeff, do you know of any other sort of Last Gift branches?
Yeah, I was just about to type in the chat. They are looking to start one in Brazil, and they've been talking to the Last Gift team. So I'm not sure which group it is in Brazil, but they're that's looking at it.
Okay.
So you can't see me, but I'm on the phone.
Oh yeah, I see you. I mean, I hear you.
But it won't let me back in, but I called in. So anyway, I'm sorry. I'm really sorry. I don't know what happened. I forgot what I was asking, but oh yeah, I know what it was. So Mathias, I mean, we have been crazed for years about getting women involved in trials and other populations like people who acquired HIV at birth or a young age, and you know exactly what you were talking about before. So I mean, that means that you're going to be looking at all of this, right? I mean, you're going to hopefully at other plagues as well. So and the things that, and I guess we can start this because it's almost time to finish up. But you know, so the intervention that you're going to use here. In other words, first of all, you just need to draw blood from people to see this stuff, right? I mean, there's no anatomical or biological reason that this shouldn't work for all human beings, right?
Absolutely, but at the same time, there seem to be differences in terms of what the immune system is able to do. In some people, it's just better, right? So that Danish patient is an example. But also, since you mentioned it in the beginning, right? Generally, the impression is that the female immune system is better than the male immune system in doing all this. That is a very clear signature. Just the elite controllers, right? In most studies, the probability to become an elite controller is 4-fold higher in women compared to men, right? So there is just that important difference related to sex that going to publish that soon. She is one of the lead investigators of the study where this is going to be looked at in great detail. So
amazing.
That's also a very exciting.
So in other words, we're going to get our dream come true, not only with something new and exciting, but that's going to include lots of other populations instead of the same old people that are easy to enroll and retain. Yeah. So the other thing is, okay, so as far as, I mean, I hate to be a nerd here, but where are we in getting this assay, the regulation process, getting it, you know what I mean? It's ready for prime time for everybody to use and getting it validated and, all those rotten hoops that something like this has to go through before it's readily accessible.
Yeah, so that's a good point. Currently, this is a research assay that cannot be broadly applied. It's also quite expensive, and it's really NIH funding and the collaboratory funding that is making it possible to use it. But we are trying to optimize it to a point that it becomes more readily accessible and can be more easily used by different centers. It could develop at one point into more like a standard assay that could be used also potentially for clinical decision making at one point in the future then. So
yeah, well, that always is the way. I mean, it gets better and easier as you go. And I guess the last important thing for me is, so what would the intervention be? I mean, is it that this travel agent? Did you aim for the travel agent to put the travel agent out of business so the HIV can't find out where to go? And where it's supposed to go and where it can do the most damage? Or, I mean, it doesn't sound like it's a hosting per se. It sounds like it's something that's targeted at the actual HIV, and that it's, the direction where it lands, or the genome, and then the genetic part where it lands.
So yeah, I mean, there's a couple of options that are being explored. I didn't have a chance to talk about it today, but one possibility would be, and this is something that we are interested in, but it is in early infancy, is to specifically change the integration size. So if HIV is sitting in a gene, okay, it would be typically likely to be expressed, but can you change that specific site in a way that HIV is repressed? So essentially inducing that block and lock position in more viruses, including the ones that are typically in highly expressed genes. That is one approach. We call this like sort of epigenetic modifications, sort of changing the ability of HIV to transcribe at the site where it's integrated in the chromosomes. So that's one possibility, and there's also certain additional approaches that are being considered.
Yes.
Michael, did you have other questions you wanted to ask? I don't want to take up the whole time.
Yeah, no. Jeff has his hand up. I wanted to give him a chance to ask it.
Yeah, remember, I can't see anything now.
That's fine. Go ahead, Jeff.
This is a logistical question. So for all these tests, do they require just a regular blood draw, or do you need large volume or leukapheresis to conduct them to get what you need?
It depends to some extent on the size of the reservoir. Some people have just a very high reservoir, and then we need relatively small amounts of cells. In certain cases, the reservoir is very low, and then we require larger numbers of cells. So in some study participants, they have been kind enough to participate in leukapheresis protocols here, specifically people who have very low reservoirs. So then we can collect like 2 billion PBMC or so. So that allows us to do a very deep analysis. But in the majority of people, it's sort of something in the range of 20 million cells or so is already very helpful and sufficient in most cases.
Yeah, nothing would prevent women per se, you know what I mean, with this leukapheresis process and little veins and all that. It wouldn't be a non-starter for women for the most part.
Well, and also doing it in more places in Africa and places like that have the capacity.
Yeah, sure.
Thank you.
Mm-hmm. So there are no more questions. I know my mind is blown.
Absolutely. Michael, did you have some questions?
No. And Linda, I just see that I'm getting you back into the room.
It's not connecting though. It's just going. It stopped even going around, I think.
Okay.
I don't know what's going on. Anyway, are there any more questions in the chat?
Nope. We're done.
Okay. Let's see. Let me see if I guess are there any other sorts of interventional things that we haven't asked you about that occur to you, Mathias?
Yeah, I mean, maybe we can, for a minute, I can talk about this sort of shock and kill approach that has been talked about a lot, right? To a large extent, it's not been particularly successful. I mean, in that Danish study, that person ended up getting romidepsin, which was designed to be a latency reversing agent. But at the same time, we believe that sort of that's not the full story, right? Because if the cells in which viral reactivation is being induced, if they are resisting immune-mediated killing just because they express surface markers that tell the immune system, just don't kill me, then these interventions are less likely to be effective. And sort of we believe we need to really go down to understand what immune mechanism is able to recognize the cells that are transcriptionally active. Once we understand that, we have a better chance to intervene in a rational way that is specifically taking advantage of that knowledge that we have generated here. So
that you're really not pushing like a shock and kill like we've talked about, though. You're just explain that to me again. I'm not sure what the point of that was.
So if you are able to induce reactivation by using some of the drugs that have been proposed and used in the past, then it might in certain cases not be sufficient, because if that cell is resistant to immune-mediated killing, if it is expressing surface markers that make that cell resistant to immune-mediated killing, then it's not likely to be successful. So we believe it has to be really a combination of intervention, reactivation of latency, or induction of viral transcription might be one component of it, but certainly not the full story. We are really just at that stage that we understand the immune system is able to do something against the reservoir. It is more effective than what we previously believed that it's doing. So that is changing things in a way that we can use that now to understand exactly what is the mechanism that's driving that immune selection process. That's where all the resources that we have are currently invested in to figure this out in greater detail.
Right.
And again, we have better technologies. That's a little bit like what I wrote in this paper that you mentioned at the beginning of the discussion there, from this HIV cure in my lifetime or so, right? That's, I believe, the stage that we stand in currently is that we understand latency is not the full story. There's additional mechanisms that are playing a role. The immune system is able to do something. So now we have a bunch of assays here, and we are better able to go deeper and understand the biology better. And that puts us into a position to develop concepts and interventions that may be more effective in going after these cells.
And also that the defective cells also play a role, because, I mean, that's extremely important because there's so many of them. If we need to, you know, if there's something you need to do to take their power away as well, I mean, that's a much bigger, you know what I mean, lift than just the active proliferating cells.
Right. So I totally agree. The cells that have defective viruses, those are important too. Even if they don't drive rebound, they can still contribute to disease and can potentially cause problems by causing immune activation and potentially other problems. So we really need to get rid of all of them. Essentially, that should be our target, right? I mean, you see all this progress in drug development and long-acting drugs and so on. This is great that these advances are made, but ultimately the aim of all this research should be that we find a way to get rid of all of the infected cells, not just the ones that have intact proviruses.
Yeah. So that's important too, because people have been saying for a while now that it's only those that, it's not the, you know, the ones that are just hanging out. It's all of them that need to be dealt with. So that's important. So do you see any role for like a gene therapy for this? Because there's so many, there's so many genes that are involved here. I mean, is there a way to short circuit it with some sort of gene therapy?
So I mean, one approach that is being tested is this CRISPR approach. So CRISPR are these technologies that can cut out certain target genes out of the genome. So there's actually a clinical trial that to my knowledge is ongoing by the CRISPR for Cure group, right? I believe that's part of your community. So,
Right.
that's an interesting and a sort of novel approach, and we're going to see what's coming out of that study. We actually are using some of our technologies here to analyze the samples that were collected from study participants in this particular clinical trial that was conducted by that BioExcision company, which is part of that CRISPR for Cure collaboratory.
Right.
So we're going to get some information. That's an exciting approach. And the second thing that is going on for gene editing is what I would call epigenetic editing, right? So that gets into the direction what I was talking before. Can you induce inhibitory genetic elements at the sites where HIV is integrated so that HIV is in a block and lock position just because you modified the DNA at that particular location where HIV integrated into? The concern is if you cut the DNA, there might be problems associated with that, right? There might be off-target effects.
Right.
The CRISPR and sort of cutting the DNA could have certain side effects and could cause problems at one point. So you might want to avoid it. That epigenetic editing approach is an alternative that avoids cutting the DNA, and at the same time, it could be repressing the virus to a point that it is blocked and locked. So that's the alternative.
Man, you're really cooking with gas today within every direction. Wow. Okay.
Yeah,
Is
so
there anything we can do for you? Go ahead.
That CRISPR project is really a new story, and it's evolving, and we are very happy to collaborate with the CRISPR for Cure team on this, and there might be an opportunity to talk about this in the future when we have more data to look into this
Right.
in greater detail. Yeah.
Right. Or Excision too, because Excision is part of that.
Yeah.
That group.
Exactly.
So, And is there anything we can do for you to help you in the last few minutes here? What can we do to help you?
I mean, getting feedback from your group is really so helpful, right? Just listening to you, hearing your opinion, your position, your impression of what we are telling you, right? That is really by itself an important part for us to understand how we can do a better job. So having occasional meetings with some of you, just learning from you and getting your perspective on this entire effort here is really helpful. And then whoever can and is interested in participating in studies, we are very open to this. And there's a number of different protocols open for inclusion. So those are the major things.
Yeah, well, we can talk about all of that stuff. That's great. Well, I don't know about everybody else, but you certainly made my day and woke me up. So this is really, on so many levels, just really, I mean, just what the doctor ordered. So thank you so much for spending your time with this, and for everybody that participated today. This has been a fabulous meeting, and we will definitely be in touch with you about the assistance points that you made. So, okay, wow. How many times did I say wow today? That's
about 15.
Anyway, thank you. Yeah, thank you, everybody, Michael, Richard, Jeff. Yay! It's been a wonderful meeting, and we'll see you hopefully on September the 10th for reservoirs and age. Which, meanwhile, we will have some big time questions that we might be able to answer from what we learned today. Okay, all right. Thank you, everybody, and we hope to see you next time.
Yes. Take care, everyone. Bye-bye.
Okay, bye.